Welcome to the Macular Society's My Macular and Me webinar, as we're now calling them. My name's Kathy Yelf and I'm the Chief Executive, and it's great to have so many people joining us again this evening. These events, as you know, if you've joined them before, are designed to bring some of the world's most eminent researchers and clinicians directly to you so that you can hear firsthand about the latest developments in research and clinical practice. So it's an interactive, in a way, session. Do feel free to put your questions into the chat function as we go along, and we'll try to answer as many questions as we possibly can during the session. Now, we particularly like, of course, to showcase some of the research work that the Macular Society has funded, and by that, of course, I mean the donors and supporters and members and the generous people who help us do this work, and you may be one of them who have supported our research projects in the past, and thank you very much indeed for doing that. And so tonight we are showcasing one of those pieces of work, and I'm delighted to say that my guest tonight is Dr. James Whiteford, a scientist and a macular researcher from Queen Mary University of London. Now, in 2019, James and his team were awarded a grant from the Macular Society for work to find a new treatment for wet age-related macular degeneration. Now, of course, it's great that there are some treatments for some types of macular disease, including wet AMD, but many of you will know that these are very costly, they're very burdensome, both for the patients and for the NHS. And of course, very sadly, they don't work for everyone. And so we need to continue to find better treatments and of course, therapies for some of the conditions that are not treatable at all now. So this is a very important and exciting piece of work. So how has Dr. Whiteford got on with his project since 2019? He is here. James, welcome. And thank you indeed for joining us this evening to tell us about the project. I'm going to hand over to you straight away, so do feel free to share your screen. I think you've got some slides, and we'll be watching the chat box, so if you've got questions, pop them in the chat function, and we'll be looking at those as they come in, and we'll come to as many of your questions as we can later on in the session. But meanwhile, James, Dr. James Whiteford, thank you very much indeed, and over to you.
Thank you so much, Kathy, for giving me this opportunity to present our research to the Macular Society. I can't stress enough how important these grants are to funding our research and helping us take things forward. So in relation to what I'd like to talk to you about today, it's really to try and explain the concept of what we've kind of discovered and how we'd like to take this forward into treating macular diseases in general. So I'm hoping really to explain the discoveries we've made and hopefully give you some insight into how we're going to take them forward. So really the title of what we do is this targeting disease off switches in macular disease. And what I hope to really do in this talk is to explain what I mean by a disease off switch. So really just to give you an overview, I'd like to introduce the team because they're the most important people in the lab. They do a lot of the work that I'm about to present, and also look briefly at new blood vessel formation and macular degeneration, and then, although Kathy's touched on it, look at some of the issues with the current therapies, and then try to explain our research, and then of course I'll happily answer any questions that you might have. So my team, we're based at the William Harvey Research Institute, and at the moment there's the four of us. We've got Dr. Samantha Oroki-Asami, Dr. Fahim Sheikh, and a PhD student, Michaela Boulderstone. And we're all focused really on working on macular disease and also other neovascular eye diseases. And we're actually based at an institute called the William Harvey Research Institute. Now, the reason why I mention this is because it is quite relevant to the work that we do in that William Harvey was a very eminent scientist in the 1500s. And he was actually the person that discovered that blood actually circulates, that we have a circulatory system. And he did this by using some fairly ingenious, for the time, experiments. But the important thing really is that he really sort of postulated the existence of blood vessels. And this is something that our group's worked on quite extensively. So the reality of that, and what our group's focused on is the biology of blood vessels and understanding how they work. And basically, a blood vessel consists of a single layer of tightly linked cells. And you can see them on this picture here. You can see that they're red on the red. And these cells are called endothelial cells. And if you can think about them as bricks in a wall, or bricks in a wall, by their nature, they have to be pretty quiescent. They have to just sit there, make sure there's no leaks, and just be an effective pipe really to channel blood around around our bodies. The other important functions that they have is we need blood vessels to transport nutrients around our bodies. We need it to bring water to our bodies. It's also for fighting infection. So obviously blood vessels carry inflammatory cells which help us fight infection. Clearly oxygen's of importance, and also to dispose of our waste. So I'm just going to stop here. So we've established blood vessels are very, very important. And one of the absolute pleasures of having been a research scientist throughout my career is I've managed to go and work in some rather nice places. One of which I spent a significant amount of time working in Denmark, around about 2007 to 2010. And I stayed very closely attached to my friends in Denmark. And one of the things I like to do over the summer is to go and help work on this summer house that they have, which is on the north coast of Zealand, which is one of the islands in Denmark. And some of the jobs we've accomplished is we've converted this area into a patio area, etc. And I find it very good to get away from the laboratory. And I'm going to use this rather gruesome example. You'll forgive me if you find this a bit unpleasant. School children love it, but I hope this audience does as well. So when I was over there doing a few jobs, I actually had a rather nasty accident, and I bumped into a nail that was in a ceiling, and I did this to my head. Now, the thing I wanted to make, the point I'm trying to make here is that obviously this is what happens when we damage blood vessels. Clearly, I've injured myself quite badly, and the blood vessels are leaking, etc. And the body's response to this type of injury is to undergo physiological wound healing. So the body kicks in and establishes a whole host of processes which help to fight off infection, stop blood, stop the bleeding, etc. But a very important process in this is a process called angiogenesis. Now, angiogenesis it occurs, and it's basically the process by which we can fix damaged blood vessels. So essentially, it's a process where it's the formation of new blood vessels from existing ones, and it's essential for our development as well as for physiological wound healing. Now, for many of us in this audience, and myself in general, angiogenesis is actually a very, very rare process. Once we've grown and developed, there's not so much the need for that active generation of new blood vessels. It is a feature of the female reproductive cycle, and like I said, also for physiological wound healing. However, an important point, and of relevance to everyone on this call, is that angiogenesis can become dysregulated. So essentially the process is no longer a sort of part of a physiological process of wound healing, but it can actually contribute to pathological disorders. And these include eye diseases, inflammatory disorders such as rheumatoid arthritis, and also new blood vessel formation is very important in cancer and in tumor formation. So I put this slide up, and again, I don't think there's very little need to dwell too much on this to this audience, but obviously our interest is trying to find, to help find treatments for wet age-related macular degeneration. And I think the really important point to make about this from this slide is that the underlying process in causing the pathology of wet AMD is choroidal neovascularization. And choroidal neovascularization is an angiogenic process. So essentially what's occurring here is angiogenesis is happening, but in a dysregulated way, and it's causing all sorts of disruption to the structure at the back of the eye. So what happens is new blood vessels emerge from the choroid membrane in the eye. These breach the retinal pigment epithelium and start disrupting the photoreceptors, etc., at the back of the eye, causing the loss in vision that we see on the previous slide. A very important point about this as well is that when new blood vessels are formed in disease states, they're actually not very good ones. They're very leaky. So another aspect is these new blood vessels are also leaking all sorts of things also into the back of the eye, which only serves to exacerbate the problem. Now, in the context of this, the major driver of angiogenesis, so this new blood vessel formation in the disease, is vascular endothelial growth factor A, VEGF-A. And I'm sure you're aware this is the target of current therapeutic targets for treating wet age-related macular degeneration. And I just wanted to put this slide up because it really makes the point actually what VEGF-A does to endothelial cells. So remember I mentioned that the importance of the key feature of an endothelial cell really is it just needs to be a brick in a wall. You don't want it to be moving, you don't want it to be proliferating. It just needs to be there to keep a solid pipe with which for blood to pass through. The thing that when an endothelial cell sees some vascular endothelial growth factor A, there's a molecule on the cell surface of this called a receptor. And in this case, it's called the VEGF receptor. And when vascular endothelial growth factor engages with this receptor, it stimulates many, many changes within the endothelial cells. And this makes them more migratory. They start to divide, they start to make more of them. And all of these processes are really geared towards making a new vessel. So if you need to make a new blood vessel, you need more cells, you need them to move, to migrate to the area that they need to be, etc. And I just put this video on purely because I really like it. And these are some endothelial cells that we've grown in the lab. And this is filmed over a 12-hour period. And what we've done is we put some vascular endothelial growth factor on them. And the effect of this is to make them do some quite weird and wonderful things as they're moving across this tissue culture dish. But it's just to really make the point that vascular endothelial growth factor switches endothelial cells from an inactive to an active state. So as Kathy alluded to, there are a number of therapies that are currently in clinical use for treating wet age-related macular degeneration. And all of these are based around targeting the vascular endothelial growth factor. So again, in a similar diagram, we have our secreted vascular endothelial growth factor here. And you can, the drugs work, they're either antibodies which recognize VEGF-A or molecules that mimic the VEGF receptor. And the basic principle is these drugs are injected into the eye, and these capture all of this VEGF and essentially inactivate it, meaning that all of these pro-angiogenic pathways within the endothelial cell are not switched on. As Kathy also, sorry, excuse me. As Kathy also alluded to, though, these drugs are actually incredibly expensive to make. And also there needs to be administration sort of several times a year, which in itself is an expense. There's a real issue with a high non-responder rate. So many patients who are prescribed this may not actually respond to these therapies. And also there's evidence now that there's a loss of efficacy after prolonged treatment and possible long-term side effects. So clearly from the basis of this, that there is a need to sort of further research angiogenesis in wet age-related macular degeneration. And this is really where our research comes in. So for many, many years, I've worked on a family of molecules called the syndicans. Now the syndicans, reside at the cell surface, so that they have an outside part which essentially is outside the cell, and an inside part which is obviously inside the cell. And in humans, we have 4 syndican family members: syndican 1, 2, 3, and 4. I think one of the most distinguishing features of them is they actually have these sugar chains attached to them. And these sugar chains are thought to modulate a great deal of the functions associated with syndicans. But I would also like to direct you to these blue regions. So if you can imagine, this is a single transmembrane spanning protein. And these blue regions I've actually called syndican ectodomain adhesion regulatory domains, and each syndican has one. And the interesting thing about these is they all exhibit the same properties in relation to angiogenesis, in that all four of these proteins actually inhibit new blood vessel formation. So I've used this example here. This is an experiment we do in the laboratory a lot to model angiogenesis. So what we've done here is we've isolated the thoracic aorta from a rat, and then we've sliced it up rather like calamari, and we can embed it into a matrix in the lab. And when we give it a pro-angiogenic stimuli, such as, oh, hold on. So I've just seen a question from John. You mentioned loss of efficacy with prolonged treatment. Can you give an idea of the length of treatment which is usually effective? That's a very, very difficult question, and I'd need to go back to the studies. I suspect it varies with from patient to patient, actually, how long the treatment is usually effective. I hope that's answered the question, John. Yeah, and
sorry, I'm muting myself. Yeah, you're right, James. There's such an individual response, and some people are very responsive and may only need a few injections, and other people really need treatment for life. And I think sort of 6 to 8 years seems to be the point at which many people start to see a drop off in efficacy.
And actually, we've got a question from Margaret as well. What are the side effects of therapies used? I mean, they can vary just from an increase in sort of ocular pressure, just from the fact that you're having the injections, to there's some evidence where that there can be damage to the retinal pigment epithelium, and I think a lot more of the, certainly from my perspective, a lot more of the side effects are evident when these anti-VEGF therapies are administered systemically in for example, a cancer scenario. In cancer scenarios, and I think there's evidence, certainly in rodent models, that would suggest that there can be adverse side effects, but it's largely when they've been administered in a systemic fashion. I hope that's answered John's question. Or not, Margaret, sorry.
Yes, again, I think that one of the problems is that obviously if you give an anti-blood vessel growth drug systemically all over the body, then you stop this healing mechanism potentially. There's a risk, and obviously people who have had strokes or heart attacks often are not given anti-VEGF treatments because we don't want to damage healthy blood vessels. But mainly it's mainly the effect of the injection that are the side effects themselves, rather than a very serious effect of the drug, unless you've got a particular risk from this particular type of medication.
Thank you, Kathy. So yeah, one of the ways in which we study angiogenesis, as I mentioned, is to grow these aortic rings. So these are rings we've sliced up from a rat aorta, and we can embed them in a matrix. And from this, we can grow these rather beautiful blood vessel structures, which is something I really find very therapeutic to do in the laboratory. But what we actually can show is that the blue regions, as I was discussing before, of the syndican proteins, all four of them actually can inhibit the formation of these. So in this experiment, the more sprouts we see emerging from an aorta, the more angiogenesis is occurring. And what we see is in all four cases, that when we've isolated these proteins and used them to see if we can inhibit angiogenesis, we actually see that we get this inhibitory effect. So we can block angiogenesis using these syndican molecules. And one of the key questions that we've been trying to answer over a number of years, and it was actually one of the projects that was actually the subject of the project that the Macular Society funded, was can we derive therapies from syndican ectodomain core proteins? So the example I'm going to use is work we did on syndican 2. Now, what we actually did, and I know this is a fairly complex experiment and not really relevant to macular disease, but it really gave us a good insight into what we're dealing with. And basically what we did is we're going to talk about one of the syndican 2. And what we did is we engineered some cells to constitutively, that means to constantly produce the extracellular part of the syndican. And what we did is we injected these into a mouse, and the idea that the cells that we have is that they're actually going to make a tumor. So when we did this, we could see that under controlled circumstances, i.e. these cells are not producing syndican 2 constantly, we got very large tumors formed. And I hope you can see on your screens, certainly I can on mine, that these were perfused with blood vessels. So obviously as a tumor developed, there's a need for it to be perfused with blood vessels. So tumor angiogenesis is a very common process within cancer. But many of the mechanisms are actually identical to what's happening in macular degeneration. What was interesting though is when we had our cells constantly producing our syndican 2 molecule, we found that the tumors were significantly reduced. And the reason for this was we blocked angiogenesis. A second example of this, and again this is just taking it on a step further. Now we did a second model where we can actually inject a gel called Matrigel. And Matrigel is full of all sorts of goodies that endothelial cells like, and it makes them want to be angiogenic, makes them want to form blood vessels. And if we inject this Matrigel into the flank of an animal as well, we can see that these plugs become perfused with blood vessels, as evidenced by the amount of blood that you actually see in the plug. And again, when we used our syndican 2 protein, we could observe that actually the blood that's perfused the plug is substantially reduced, if not negligible. So what these experiments have told us is we've actually, in the case of syndican 2, we've actually identified an anti-angiogenic molecule. And as a molecular biologist, one of the things that we always like to do is to find which part of the molecule is important and which part is the active site. And one of the things we were able to do was to establish that this small part of the protein was actually the business end of this molecule. So what we actually did was we generated what's known as a peptide. Now, again, forgive me because I know we've probably got quite a broad audience. So a protein consists of a sequence of what's known as amino acids. So essentially these are these letters here. Now, what we wanted to do was to try to miniaturize our molecule as much as possible for various reasons. And so what we were able to identify is the green region, the business end of the syndican 2 that we know is important in inhibiting angiogenesis. We actually made a discrete molecule called a peptide based on the human sequence. So the human part of syndican 2 that has the anti-angiogenic properties, we actually got synthesized by a company. And this compound we called QM107. QM107 stands for Queen Mary. So, Rosemary, that's a really good question. The reason why I focused on the syndican 2 story is I wanted to try and get the concepts across. And actually the work that we've done as funded by the Macular Society has found that a combination between the molecules we found from syndican 2 and the molecule we found from syndican 3 is the most effective. But I wanted to try to get across this concept of disease off switches, and I realized I only had half an hour. So I've only expanded on well, essentially our lead candidate as things stand. I hope that's okay. So this initial developmental candidate was called Queen Mary 107. Now, when I started on this, we actually filed a patent, and that they wanted to give it more of a corporate identity, hence the Queen Mary tag. But this was quite a new initiative, and it would have been called Queen Mary 1, but we didn't feel that was very, we didn't really feel that it didn't really make it sound like we had too many molecules. So I said, so they said, what about 100? And I said, well, 7's a lucky number, so let's call it QM107. But anyway, one of the key issues with this molecule, we then wanted to test it and ask the question, could it potentially be effective in treating wet age-related macular degeneration? So we did this experiment. Now, you'll recall from when I was describing how new blood vessels form in the eye during wet AMD. And I mentioned that blood vessels emerge from the choroid layer in the eye. So we were able to model this in the laboratory. So we were able to isolate the choroid layer from rodent eyes. And rather like the aortic ring model, we can we can place these explants into a matrix and we can grow blood vessels from them. The more blood vessels you see, the greater, the more angiogenesis is occurring. And what we saw when we treated our explants with our QM107, we found we could substantially inhibit angiogenic new blood vessel formation from this, and this could be achieved in a dose-dependent way. So we then reached out to colleagues at the Institute of Ophthalmology at UCL, and we performed what in many regards is perceived as the we'll wait to the end of the one from Claire there. What we actually did with colleagues at the Institute of Ophthalmology at UCL is we performed what's widely regarded as a leading preclinical model of wet age-related macular degeneration. And this is actually using a laser to induce choroidal neovascularization in mouse eyes. And essentially what it entails is we apply 3 laser burns to the back of a mouse's eye. And what you can see from these angiographs is that you get the formation of these lesions in response to the laser burn. And the size of these lesions reflects the level of the choroidal neovascularization, of the angiogenesis that's occurring in the eye in response to that. And this recapitulates many of the features of wet AMD. What we were very gratified to see, though, is that when we applied these laser burns, and if we then also inject some of our QM107, we found we could inhibit lesion formation drastically, as also evidenced by these red lines on the graph, indicating that these peptide molecules could actually work. Well, it's all very well doing things in test tubes or on cell cultures, but this is actually inside an animal's eye, and we're seeing that we're getting a therapeutic effect. So this really is kind of what we've been trying to do from develop from the Synergan family, is these types of molecules which we can inhibit new blood vessel formation in models of wet age-related macular degeneration. Now, I wanted to go back really to explain really what we think we've discovered and why I think this is probably important, and it will inform our research moving forward and could actually be even more powerful than we actually think, than we actually feel right now. It's a really important point to make, is that during physiology, if we assume that the aberrant angiogenesis we see in wet age-related macular degeneration is essentially a feature of wound healing that's gone wrong, it's become dysregulated. One of the things about physiological wound healing is angiogenesis stops. So when I cut my head open, the healing process is kicked in, new blood vessels formed, etc. But there had to be something within my tissue that said stop. I.e., in as much as you need wound healing to occur, you also need to have a signal to say, okay, wound healing's finished. We're happy that the wound is, the tissue is back to normal. And what we've established is that our peptide is actually stimulating what we call a disease off switch. So what we think we've stumbled across is like the body's natural way of switching off wound healing, which we're kind of suggesting we could use to switch off aberrant wound healing processes in macular disease as well. So what happens is QM107 actually interacts with another cell surface receptor called CD148. And what this actually acts to do, it switches off all of the pathways that you see switched on by VEGF-A, it actually switches off. So the net effect of that is to tell your endothelial cells to stop being active and angiogenic and to go back to being just bricks in a wall, just being part of a blood vessel. And if we compare this approach to anti-VEGF therapy, I know I'm very biased here, and I may, obviously I would be because it's what we work on, but I do think targeting an endogenous off switch on an endothelial cell in a disease state is likely to be more effective than targeting a secreted factor where you still have the on switch available. So this is really the key distinction about the discoveries that we've made, is that we've actually discovered a way of turning angiogenesis off, which is distinct to anti-VEGF therapy. And really, I'm going to leave it there. I could tell you about so much more, but Kathy did say it was only half an hour, and I do find when I'm speaking online, I tend to go on and on and on. So I'm just going to take you really through the next step. So certainly for QM107, we've actually tested and shown it has efficacy in other molecules of neovascular eye disease, such as diabetic retinopathy. One of the things you have to bear in mind as well about these peptides is they're actually very small molecules, and the MHRA actually classifies them as small molecules, which is very useful. We felt that because of the size of them, and also actually they're actually, because it's a chemical synthesis, in comparison to anti-VEGF therapy, that it's incredibly cheap to make. So we've explored whether we could actually use alternative modes of delivery, such as eye drops. The complexities of this, I'm sure many of you will also be aware how challenging that is. But certainly in rodent models, we're pretty confident we can achieve penetrance with these molecules into the rodent's eye. Obviously, we can't ignore the cancer data, but we're not really in a position to explore that. And finally, and these discoveries are perhaps most excitingly of all, that we're currently under negotiations, although they're proving quite lengthy, with an ophthalmology company called Oculus. And basically, we've actually, there's the patent that I mentioned previously, but the work we've done with the Macular Society in terms of the combination therapy, that was also the subject of a patent filing last year. And we're just exploring a licensing opportunity to see if they'll actually license it. Because really what we need to take this forward now is actually the preclinical toxicology to see if we can actually try a phase 1 or a phase 2 trial of this. So that's what I'm trying to, get the next stage of funding really to take our discoveries forward. So thank you, and I'd happily answer any questions.
James, brilliant. That's absolutely fabulous. There's some questions coming in already, and in fact, one of them actually is asking the question that was actually on top of my list about how to ask you. So we know that the current treatment for wet AMD is an intravitreal injection, that is to say an injection that's given actually into the eyeball. Just to reassure everybody, you don't see the needle coming through the pupil, you look to one side and it's slipped into the corner of the eye and your eye is numbed and you don't feel it. So it sounds much worse than it usually is for most people. What would be the delivery mechanism for your peptide? Because we have got this problem of the systemic problem of putting this off switch all over the body could be quite a problem, wouldn't it? So would your peptide have to be delivered directly to the macular area, or would it go in some other way?
So Kathy, in relation to that question, so we're very keen to try to translate this into some sort of human trial, and we felt that the simplest way to do this would be to follow the existing route of administration, because we felt that it takes one less complication out of achieving what we want to achieve. And we then felt that, well, if we got through to the point where we've trialed this in man, we're showing efficacious benefits, etc., then alternative routes of administration could be explored. But we just felt dealing with the MHRA with a new compound, etc., we thought if the way we envisage it at the moment, it would be administered in the same way as anti-VEGF therapy. But it was more just to simplify that to simplify our job, really.
Yeah, no, I understand that.
So in the future, I mean, the problem is with the eye drops is ensuring an accurate dose. You know, from how do you administer it to make sure you get an accurate dose, etc. And the other thing I should mention that I had a very, I'm not sure if any of you watched the program Dragon's Den. I did a pitch to a sort of venture capital thing, trying to get investment in. And you know, when the panel gets quite hostile, they were very, very hostile to the eye drops idea. And particularly for the US market, because apparently the clinicians get a fee for every injection they give, and the argument was that they wouldn't ever prescribe Cure 107 because they wouldn't get a fee if it was through eye drops, etc. So it was a chastening experience, but every time I've had these, I've learned. So yes.
Yes, that's a rather depressing outcome, isn't it, from that conversation? I mean, you're right about eye drops are difficult because, of course, the eye is structured and it's a beautifully created organ, isn't it, that has got so many defences to stop foreign material getting from the front of the eye to the back of the eye. So it's quite a challenge anyway, isn't it, to deliver something as an eye drop to the retina. Okay, for glaucoma, conditions that are front of the eye conditions, but the back of the eye is much more challenging.
Yeah, so we do. There is one of the interests actually that Oculus has is in glaucoma, because it seems we've actually stumbled across these disease off switches for a number of repair mechanisms. So not only new blood vessel formation, but also fibrosis as well. Mm. And so it seems that this switch seems to switch an awful lot of stuff off that's related to these processes. So certainly, I think that would be another interest for. Certainly for Oculus anyway.
So there's a question here. So would it work for other choroidal vascularization diseases, such as myopic maculopathy or some of these other inherited retinal conditions that where you see new blood vessel growth and so on?
Without testing them, my hunch would be, so long as CD148 is there, then we can. That's what's going to help switch off the pro-angiogenic mechanisms. I think it's also quite interesting that we've also just recently, and again, it's kind of really the end point of the project that we've had funded from you. We can actually block drusen formation with these peptides. And we were really, really excited by this, but we kind of need more. This isn't a plea, but we kind of need, you know, it's kind of going to be the next grant really, because it seems to be able to block the production of this in this model. So I think that's potentially very interesting as well. But we're so again, that's something else that we'd like to explore.
Yeah, but yes, I mean that would be very interesting, because the holy grail of this, really, I mean, when you get to wet AMD, then you have disease that is, you know, considered advanced disease, isn't it? And with all health conditions, the aim is to try to prevent the escalation of the condition to get to an advanced stage. The earlier you can pick it up, the more sight that we'll be able to save. I just want to explore a bit more, a couple of other questions which we'll come to in a second, but just to go back to this, you mentioned how the growth of new blood vessels is important in cancer, for example, and there are going to be lots of conditions where this is the case. So the first drug that was developed for wet AMD was, of course, a repurposed cancer drug, wasn't it? It was originally developed for cancer to prevent or to slow down the development of the blood vessels that would grow around a tumor to support it, like it would support an organ effectively. So switching this off switch could have quite a broad applicability.
Yes, absolutely. I mean, we another aspect of the work that we've kind of we have a very good colleague in the US who works on fibrosis, and he's very keen on utilizing this technology for treating fibrotic disease, which is it's a huge problem. But absolutely, I mean, there are definitely other indications. One of the reasons we focused actually on ophthalmology, and it's probably quite clear from my presentation, I'm not a trained ophthalmologist, but we found that in order to prove that these things actually block angiogenesis. The eye is such a wonderfully tractable target because we didn't have to worry about losing the peptide in the system, or we knew that we can inject it and it's there. If it blocks angiogenesis, it does. But now we've since done, you know, I think we've done an awful lot of stability type work with the peptides to establish whether it would actually be systemic, work systemically. So yeah, the thing, I mean, for us, we don't have the. I'm going to be absolutely honest with you that my groups, we don't like the cancer models, or some of the work that goes on with the animals, etc. We would prefer just to stay working with the eye.
Yes, I mean, lots of people really would prefer to do that, wouldn't they? And I guess if you're going to do, if the next stage of this would be to test whether there's any toxicity, whether the side effects of it would be damaging, what would be the next stage then of how you would go about doing?
Well, actually, so yes, I mean, we've actually done quite a lot of work in the laboratory already. We had a meeting with the MHRA just before the pandemic. And kind of establish what we need to do to actually establish that this will be safe for humans. So what's really very reassuring is that these molecules, they don't generate, the immune response they generate is absolutely negligible. They don't provoke any inflammatory response, and as far as we can tell, cells can tolerate extraordinarily high doses of them with no adverse effects. The one thing that you should bear in mind is that these molecules are derived from receptors that we actually express in our bodies anyway. So our bodies are used to seeing them. So one would anticipate and be hopeful that the toxicity profile and experiments that we need to do are, you know, they would come out favorably. And one would hope that the side effect profile would be negligible.
So once you've done the toxicology work, and then you'd be looking at full clinical trial.
Yeah.
And would that be kind of testing the dosage levels first, or you'd start with a small number of patients and build them through the various phases of the clinical trials?
One of the key steps actually, and it would be at the moment we've only worked on rodent models, and we need to do some level of work in larger mammals. So, for example, the MHRA has suggested that we do the bulk of the work in rabbits and then a small amount of non-human primate work, which, although is upsetting, you have to understand that only primates have maculars, and we're working on macular degeneration. So that's been minimized, but that would also have to occur. And I think in that respect, we would get a very good steer as to the dose that we would require to use in humans based on just the volume of the eyes. I mean, you know, a mouse eye is tiny compared to a rabbit's eye, compared to a human eye. So I think we would have a much clearer, once the body of work that we're proposing to get done, once we've done that, then we would have a very much clearer idea of the dosage. But I would also stress that the actual active amount that we use is minuscule. It's about 1,000-fold. It's about 1,000-fold less than you would use for anti-VEGF therapy in terms of the actual amount of the molecule that you need. And I think actually partly this is due to the fact that we're targeting these off switches, as opposed to trying to, by actually targeting the cells and not the secreted molecule. I think it's much easier to achieve the therapeutic benefit.
So it's fascinating. Questions come in, and every time this is the $10 billion question, really. What's the time scale for this sort of work? I mean, we know that clinical trials take a minimum of 2 years, don't they? And then you've got the data crunching and all that kind of thing. And you've probably got 3 stages of clinical trials. You've got the preclinical work to do and the toxicology reassurances to do and so on. So, you know, this is not just around the corner, is it, to be
so the challenge for me as a university academic is to try and get the funding and secure this. I mean, unfortunately, the speed with which this can be done is actually down to how fast I can raise the grant funding or whatever. If we can engage if we're successful with the licensing deal with Oculus, they'll have the mechanism and the resources to really push this through quite quickly, at least to the point where we would maybe have got through to phase 1. And the kind of timeline project that that's under discussion, you're looking at maybe well, 18 months to 2 years if everything goes to plan. But they just have to agree to finally just sign the contract and we get this done. And then moving beyond that, I mean, yeah, obviously the nature of the clinical trial, etc. That again is why I need help actually from. I'll hold my hands up. I mean, we would need help from Oculus and the clinicians to actually on the design of that, the length of exposure to the length of the treatment, and also recognizing what is a good and bad outcome. So I think in reality, based on, as I said now, I think I can get this to phase 1 clinical trials, but beyond that, we'll need help from either Moorfields, the Institute of Ophthalmology, etc., or indeed a company like Oculus.
Yeah, well, I mean, this is why in the end, we all as small funders and as academic researchers need industry partners to come in and help us actually turn this properly into medicine, don't we? I mean, you know, it's very difficult to do it, and the costs, of course, are absolutely huge. Question here from wet AMD and CNV, choroidal neovascularization around the macula. Does it only occur in the macula? I think this is the question. Does it only occur around the macula? And if not, why not? Is that the question? I'm not sure I fully understand it, but I think it's to do with where CNV occurs in macula.
Yeah, so my understanding is it's kind of, I think as it progresses, that more areas will be affected, but my understanding is that that's where the kind of focus is, that's where the particularly the angiogenic phase actually really starts. So yes, I mean, it's an interesting thought that, and there's a number of other areas in angiogenesis research is why some tissues don't have blood vessels and other tissues do, and you could almost argue, I mean, the eye is avascular pretty much once you're so you know what signals are there that stop this occurring, etc. So I think, yeah, the localization of where the angiogenesis starts is probably a really important question.
And I think that's one of the problems with understanding the root causes of AMD, isn't it? That we don't really know where it starts. Where in the structure of the eye does the problem first begin to emerge? Drusen may not be the first sign. They may be a response to something that's happened.
Well, I mean, an interesting study, but it would be something I'd need to, an interesting study would be to look at in early stage patients, what's happening with CD148, the disease off switch. Because certainly in lung fibrosis patients, you see a very marked loss in the off switch early in the disease, which seems to promote or enable this aberrant wound healing process to occur. So it could be even even some sort of diagnostic element to this, but again, it's quite a big statement saying we've discovered the off switch for wound healing. I think we need to expand upon this more.
There's a question about whether any of this is applicable for dry AMD. Of course, that's angiogenesis is not a feature of dry AMD, but nevertheless, there are disease processes going on there, aren't there, that also presumably have off switches?
Yes, and I think it's something that we'd like to explore further. It certainly does seem that these molecules have a way of making cells behave themselves essentially, or act as you'd want them to. So again, it would be something we should try and investigate.
So thank you very much for that. So just going back to a question that came in while you were talking, which was about BioView. So BioView is one of the new treatments for wet macular degeneration, and it's an anti-VEGF drug, and there was a little bit of controversy about it when it was launched, because it was thought that one of the side effects was inflammation, but also this point vascular occlusion. And the question is whatever that is. So I don't know what your knowledge is of Beoview. I know you're not a clinician. You might not have had a great deal to do with Beoview, but certainly vascular occlusion is just a blockage in the vein, isn't it? That's
yeah. I think one of the issues with this, and I think it goes back to a point I made in the talk, is that the new vessels that are made in disease states such as macular degeneration, that they really are not very good ones. They're not very organized, they're unstructured, they're leaky, and it's conceivable that one of the adverse effects is essentially you're almost tying these vessels in knots almost by with the blocking VEGF, because so certainly I could envisage a situation where you would get blocked vessels within that. I'm not sure really how extensive that would be with Beoview.
Well, I think it's marginal. I think that the company has said that it's not significant, and it certainly was licensed, and it's been given a approval by the Medicines and Healthcare Regulatory Agency and all the regulators. So it's not regarded as an insuperable problem. But that's what it is. Vascular occlusion is a blockage in a vein, and I think it can happen. All the anti-VEGFs have some side effects. No drug is ever side effect free, I guess, is it? But the advantage of Beoview is that it lasts a bit longer in the eye than some of the other earlier anti-VEGF drugs, and that's why people like it, because it stretches the time in between injections.
I mean, certainly that's a factor for us as well. I mean, one would hope that if the hypothesis is true that we've got these disease off switches, one would hope that actually the frequency of administration would be significantly less as well. I mean, certainly in the experiments that we've been allowed to do, we found a very long-lasting positive therapeutic benefit in the length of time that we've been allowed to do the experiments. But we're talking weeks, not months, and really months is where we need to be with this.
Yeah. So the grant that you got from the Macular Society was, in the great scheme of things, pretty modest, wasn't it? But you seem to have done a huge amount with it. So many congratulations.
We'd like to give bang for our buck.
Well, I think you certainly have. So you'd be looking now for the next stage of funding, and I'm sure, I hope that you'd come back for some follow-on funding from us.
Well, yes, because there is the June 10th. We are working towards that. But I mean, like I said, hopefully, if we can just get lucky with this company there, I think that's going to really boost things. And I was also, we've also put in for a substantial amount of money from the MRC in January, which we're going to hear about very soon, which would be a real, again, that will be a real boost to things. On a slightly, it's another syndicant, but it's on a similar sort of pathway. So hopefully, yeah, we'll get some good luck with that.
To what extent were you affected by COVID and lockdowns and things like that? What impact did that have on your work?
I mean, that's a whole different. So what happened? They actually shut our institute from the March, whenever it was, through till about June, and I foolishly volunteered to go and start a testing lab up in Milton Keynes. So I was one of the first people up there doing well, literally the first person up there trying to set up a testing laboratory. And we went from doing 7 tests on the first day to, after 3 months, we did about 20,000 or something like that. So that kind of significantly affected my sleep during the pandemic, but thankfully I was busy. But then we were very blessed in that the laboratories were open again in June, and we had this rather crazy system of shifts. So I was doing the morning, and my colleague who's done pretty much all of the work for Macular, she would come in the afternoon. We'd sort of be doing almost tag teaming on the experiments because we had so much of the social distancing. So in many respects, it was actually, from a work perspective, very enjoyable, because there's so many other aspects of university life that we didn't have to deal with, and we could just focus on the science. So in a way, it was very productive, but I think all of us would have been happier if it hadn't have happened, really.
One of the things that I think a lot of people have expressed frustration with macular research and how difficult it is to get funding for it and so on, and seeing how when enough people throw enough money and enough effort at a problem like COVID-19,
oh yeah,
actually you can turn around a new vaccine in a year, and people are going, well, if you could do it for COVID, why can't you do it for macular degeneration? It's infuriating, isn't it, that it's so difficult to get funding in this area.
I think it's also targeting the resources in the correct way. I think you can be incredibly efficient with the resources that you get. I mean, but I could argue that, I mean, that's also within our institution. It's you know, we'd love to get the gear to do laser CMV here, but they just don't see it as being a priority. But then we could just be on the end of a huge licensing deal from the university, so it should be a priority. Of course it should. So yeah, I mean, yes, you're absolutely right. I mean, the amount of, I mean, actually the biggest frustration from the COVID thing for us has been getting hold of things that we actually need for the laboratory, because everything was put towards testing. So all the bits of plastic that we use and all those things, and then the price of gloves, it went from sort of £7 a box to 45. You know, I mean, that's been a frustration, but hopefully, with luck, we're through the worst of it now, and hopefully that the funders as well will focus on things other than COVID.
Well, we wish you all the very best with it, James. It's been a lovely, fascinating, as so many people have said in the chat, interesting talk this evening. Thank you so much for your time and
it's been a pleasure.
The benefit of your insight and expertise, and we wish you all the very best with the next stage of the work, and more power to your switching off elbow. We hope that it's all a really great success, and we'll follow it with a lot of interest, James, and I hope you'll come back and tell us about the next stage of it. That we'd be very keen to hear how you're progressing. So thank you very much indeed, James Whiteford.
Thank you, everyone. A pleasure to meet you, albeit virtually. So thank you.
Thank you very much indeed. So thank you to everybody for joining us this evening. Don't forget that this talk will be on our website in a couple of days' time. And if you want to see any of it again, then of course you can go on to the website and look at that. But if you know of anybody else who you think might be interested in this talk, do please signpost them to it and tell them where they can find it. Next month, My Macular and Me will be on the 17th of May, Tuesday the 17th of May, at the usual time of 7 o'clock. And look out on the website for news about who our guest speaker will be on the 17th of May. But meanwhile, James, thank you very, very much indeed for joining us. Thank you everybody for joining us this evening, and have a very good rest of the evening. Thank you very much indeed. Good night, everybody. Good night.
Good night.